Okay, I'm going to demo QTLNetMiner now. So QTLNetMiner has a Google-like search interface, which is here. It allows users to specify QTL region and to enter a gene ID list into this box here. So I'm going to load an example query, which will fill this query with some keywords that are related to a particular trait, with 2 QTL regions and a list of wheat gene IDs. I'm now going to press the search button and jump into the results view at the bottom of a page. So we will have 4 different views to look at the data. The first view is called the gene view, which contains the list of genes that were related to the keywords that the user entered. In the map view, we see the wheat chromosomes and can see that only genes that are within those 2 QTL regions that the user specified are highlighted. The evidence view shows us all documents that contain the keywords, and I'll come to the network view later. So back to the gene view for now. The table is sorted by a score that QTLNetMiner calculates. The user column tells us if the gene was within the user gene list, and the QTL column tells us if the gene was within a QTL region that the user specified. So we have one particular gene here that is on top of our list and is within the gene list and also within the QTL region. And at the same time, it has lots of supportive information that tells us that this gene is somehow related to the trait descriptions that the user used. What those different symbols mean can be looked up in this legend here at the bottom of a page. So we've got phenotypic descriptions, we've got Go terms and trait ontology terms, publications, pathway information. So we can, for example, click on one particular evidence type and it will tell us what sort of concepts were found that contain the keywords that the user used. So for in-depth investigation to see how was this gene actually connected to this particular evidence types, we have to go into the network view. To see the gene evidence network, I can either click on one particular gene, or I can select multiple genes from here and then click on Show Network. For now, I'm just going to click on one gene and jump into the network view. Alright, so the structure of this network is a directed and labeled multigraph. Every node and every edge in this network has a specific type and a biological meaning. So to really understand why particular genes were related to those evidence documents that I showed in the gene view, we have now to go and look into the nodes and edges and see actually what they mean. To do this, I'm going to open the item information window and now start on the left hand side by clicking on the gene which has the flag. This is the gene that I selected earlier in the gene view. So we see lots of information about the gene. We see first the type tells us it's a gene. Then we've got IDs, accessions, the tax ID, start, chromosome, other attributes that go with that gene, all shown in the gene view information view window. So now by clicking onto an edge, we can see the type of the edge, which is here on the top and says this gene encodes, encode is the type, encodes this protein. The protein sequence is given here. So we can now continue and click on the next edge on this red one, for example, which will tell us this is an ortholog relationship that connects the wheat protein to an Arabidopsis protein, and the sequence identity is an attribute of the edge, which is 78% in this case. So, of course, this must be now the Arabidopsis protein. The synonyms and names are shown here, different IDs are shown there, and the sequence is at the bottom. So we keep following the path and studying what the meaning of the different edges are. So this edge is telling us that the protein, Arabidopsis protein, is actually an enzyme, and this enzyme is catalyzing a particular reaction, and this reaction is part of a particular pathway. This is all information that is coming from RSpsych. So basically we followed one particular path now that showed us that this gene is related to the keywords that the user used in the beginning. So there are lots of different types of paths that connect this gene now to different types of evidence. So for example, there are lots of paths that go and end up in these orange rectangles, which are publications, and every publication will contain the keywords that the user used in the beginning. And it's up to the user to decide if he really trusts this path and if the edges are strong enough so he believes that the information that he sees is true, or it might potentially be a false positive. Okay, I'm now showing just a couple of features that the network viewer has. We can easily hide information by going right-clicking on nodes and then saying hide, same concept class, which will hide now everything that was a publication. We can go to layouts and relay out the network. We have now hidden publications, so the network is a bit clearer to read. And what we can now see, for example, is that there is a path that connects this gene going this way over here to this particular node here. If we zoom in now, we can see that this is a trait ontology term called seed color. And the way this trait ontology term was connected to this particular Arabidopsis gene, which is in CHS coding gene, is through this relation. And this relation is based on text mining. So what we've done when we created the pre-integrated networks is we mined the literature for occurrences of trait ontology terms and gene names and created links between them with scores that show how strong the association is. So what the user sees in this evidence network is really just the type of evidence documents that contain the keywords and that have a valid path connecting them back to the wheat genes. There is still lots of information that is actually not shown in the first view, but can be displayed if the user is actually interested to know more about genes, even to see things that might not be directly related to his query. So he can right-click on something and say, show me the neighborhood, show me, for example, which biological processes this particular Arabidopsis gene is annotated to. By clicking on it, he gets all the Go terms that this gene was annotated to, and what he can do now is click on particular Go terms. We've got this annotation here, which is a description, and synonyms. The Go ID is shown there. The user can actually click on an edge and he will see the evidence types For example, this one is inferred from reviewed computational analysis. They are part of the evidence attributes. Okay, so this is QTLNetMiner. It has many more functionality, but I hope this demo could demonstrate the most powerful features of QTLNetMiner, and I hope that you can find it useful.